Well, you might have heard in the popular press, popular media, the newspapers about this crazy idea known as intelligent design. What I want to do over the course of our presentation this evening is to hopefully dispel some of the common misconceptions that you might have surrounding intelligent design, respond to some of the objections, and to clarify and illuminate some of the common stereotypes that abound in relation to intelligent design. But before I get there, let me just say one or two words about what I do in my ministry. As we just heard, I'm the founder of the Apologetics Academy. This is the website up here, apologetics-academy.org. And every Saturday I bring in, it's every Saturday at 8 p.m., I bring in a different scholar or expert or apologist to talk to us about a topic of interest to Christians, particularly topics that pertain to the question of whether Christianity is actually true. And I bring in not just Christian scholars, but also non-Christian skeptics, atheists, Muslims, agnostics, to tell us why they don't believe Christianity to be true. I operate with the philosophy that if Christianity is true, which it is, we don't have anything to fear from rational skepticism or critical scrutiny or honest questioning. And so following the presentation, then we have a time of interactive dialogue, debate, discussion, and Q&A. And we determine whether the arguments against Christianity really stand up. So it provides opportunity for Christians to have access to a scholar they might not otherwise have access to when they bring on Christians. And when they bring on non-Christian skeptics, it allows Christians to practice their skills in debate and also allows the less experienced Christians to see how well those antagonistic ideas actually stand up. You can also email me at this address, and that's also my public Facebook page. We also provide a service for doubting Christians. People can contact me via my website and indicate that they are struggling and wrestling with real, sincere intellectual doubts. And I'll set up a panel discussion to discuss their doubts in confidence and talk about whether their reasons for having doubts are actually really reasonable and rational. So let me just open, since I'm talking in a church this evening, with a passage from Scripture, Romans 1, 18 through 20. The wrath of God is being revealed from heaven against all the godlessness and wickedness of people who suppress the truth by their wickedness, since what may be known about God is plain to them, because God has made it plain to them. For since the creation of the world, God's invisible qualities, his eternal power and divine nature, have been clearly seen, being understood from what has been made, so that people are without excuse. The Apostle Paul wrote this in the 1st century AD. As an undergraduate student studying the wonders of the cell and the engineering prowess of molecular machines in the cell and the information storage and retrieval mechanisms that we find run the show in biology, I was filled with a tremendous sense that if that was true in Paul's day, in the 1st century, how much more true is it today in light of modern advances in both the physical and life sciences? In fact, it often baffled me as an undergraduate student, and also to an even greater extent as a postgraduate student, at how anyone could go through a 4-year university or college program in the sciences and come out an atheist at the other end, because the evidence seemed to me to be so compelling for the truth of theism. So let me just give an overview of what we're going to be discussing over the course of this first presentation. We'll begin by introducing the concept of intelligent design and define what actually we mean by intelligent design, and just as importantly, what we don't mean by intelligent design. We'll explore the implications of the information-bearing properties of DNA. We'll look at molecular machines in the cell and why that's significant to the question of design. We'll look at some of the popular and common objections to intelligent design and how we might address those objections. And finally, we'll draw some conclusions. And then in the second presentation, after a short break, we will demolish Darwinism. So let's begin by introducing the concept of intelligent design. So what is intelligent design? What is this idea that we call intelligent design? Well, there are different definitions that abound, but one popular definition that's proposed and promulgated by the popular press and the media and the newspapers is the following definition. Intelligent design is the idea that living systems are too complex and improbable to be explained by the process of evolution, and therefore require invocation of a supernatural being to explain their existence, such as God. Let's have a show of hands. How many people would go with this definition of intelligent design? How many think this is a reasonable definition of ID? Yeah, quite a few of you. Okay. Anyone see any problems with this? There's a serious problem with this definition of design. It's the God of the gaps objection. It's based upon not what we do know about biology, about science, but what we don't know. It's using intelligent design or God or supernatural agent as a placeholder for that of which we are ignorant. We can't explain feature X, so we'll just plug in God or an intelligent design or a supernatural being until we find a better explanation. And as science advances and these holes in our knowledge are filled in, then intelligent design has to retreat with each new advance in scientific discovery. There's the God of the gaps argument, an argument from ignorance. Here's a better definition, which I think gets around this problem, where I frame intelligent design as a positive inferential argument. Intelligent design is the idea that we can infer from patterns in nature that some features of living systems are best explained by an intelligent cause rather than a stochastic process involving chance and necessity. So right away you can see that by framing it as an inference to the best explanation, I have rendered it a positive argument based on what we do know about the natural world rather than what we don't know. So that's a key difference, and it's important. Now, what's the competing contender as an explanation for life's origins? Let me introduce to you the conventional view of evolutionary theory. This, of course, being an illustration from Charles Darwin, which represents the tree of life. And the idea is that all living forms alive today are descended with modification from previous ancestors. And eventually, if you run the tape of life back far enough, all life comes back to a single universal progenitor, a last universal common ancestor that lived approximately 3 or so billion years ago. Now, the theory of evolution consists of 6 main elements or components. We have the change in allele frequencies in a population over time. An allele, for those of you who are not biologists, is a gene variant. And there's different mechanisms that result in the change in frequencies of alleles in a population, including forces such as genetic drift and natural selection, and so on. There's the process of speciation, the idea that there perhaps are geographical barriers that isolate different populations, and eventually they give rise to daughter species, and you get new species. And that's a concept also associated with modern evolutionary theory. Then there's the proposition of common ancestry. The idea, as I mentioned before, that all life forms alive today are descended with modification from previous ancestors. Then there's the idea of gradualism, the idea that evolution does not happen by sudden leaps and bounds, but it happens very gradually and slowly. There's the idea of natural selection. Natural selection preserves the successful combinations of gene variants, those that help the organism survive and reproduce and produce offspring, are selected for and go on to become dominant in the population. And then there's the idea of random genetic mutation. As the DNA is being copied, the DNA polymerase occasionally makes an error, makes a typing error, if you will. And that's called a genetic mutation. And sometimes most of the time mutations are neutral. Sometimes they are harmful, and very rarely they are beneficial. And if they're beneficial, then they can provide the grist for the mill of natural selection. Natural selection can select them because they provide an advantage to the organism to survive in its environment. Intelligent design really doesn't challenge this first one, doesn't challenge the idea that allele frequencies in a population change over time. Intelligent design does not challenge the idea of speciation. Intelligent design doesn't even really challenge common ancestry. Intelligent design proponents indeed disagree about the extent to which organisms are related to one another. For example, Dr. Michael Behe at Lehigh University in Pennsylvania, a well-known intelligent design proponent, subscribes to the proposition of common ancestry. And there are other intelligent design proponents who would disagree with common ancestry. You could have guided evolution, guided by an intelligent agency. Then intelligent design is not necessarily incompatible with gradualism, although it doesn't commit one to gradualism. Intelligent design is compatible with the idea of natural selection. Indeed, natural selection happens. We observe antibiotic resistance, insecticide resistance, and so forth. The only one that intelligent design really challenges is the last one, the causal efficacy or sufficiency or adequacy of random genetic mutations to provide the variations necessary to generate the complexity of life that we see today. How do we detect design in the world? Here we see a cliff face. How many of you would recognize that this is the product of design? Not very many. This, of course, is a very complex pattern. It's very improbable, and yet we know this is generated by wind and erosion. What about this cliff face? This is South Dakota, Mount Rushmore, where you see the faces of various US presidents carved into the rock, and immediately we infer that it's the product of design. But this cliff face is no more improbable than this one. They're both very, very improbable conformations. Nonetheless, we know that this one is the product of design, because it's not only improbable, but it conforms to some independently given pattern that we recognize, namely that there were various presidents with those faces, and someone has taken the trouble to chisel these faces into this cliff face. Some of you might have seen the movie Contact, starring Jodie Foster. This is based upon the search for extraterrestrial intelligence. It's based upon a book by a novel by Carl Sagan, the famous popular science writer and cosmologist. And basically the idea is that the SETI researchers discovered radio signals from outer space corresponding to the first 20 prime numbers and inferred that they were observing not stochastic noise, but some extraterrestrial civilization was attempting to communicate with Earth. And so they were able to infer design, the product of design, by virtue of not just the improbability, but also the specification, its conformity to some independently given pattern. Here's another example. Which of the following is design? Notice they're both 30 characters. They're both equally complex. They're both equally improbable. Nonetheless, we recognize that this last one is the product of design. And beginning was the word, obviously taken from the first verse of John's Gospel. Whereas this one is simply a random array of characters on a keyboard, and so could be produced by random typing errors. I want to introduce at this point to you the concept of specified complexity, which is how we infer design in our everyday experience. Specified complexity can be describes phenomena where two criteria are met. Number one, the probability of a phenomenon being produced by chance alone exhausts the available probabilistic resources. And two, the phenomena conforms to some independently specified pattern. If you don't understand this yet, let me give you an illustration. Here's an ATM machine. We've all used ATM machines. And you put in your bank card and you dial a 4-digit number. And there's only one PIN number which is specific to your bank card. Now, if you have just 3 trials before the bank locks you out of your account, that corresponds to the probabilistic resources, the number of opportunities you have to get it right. Now, there's, on a 10-digit PIN pad, there's 10,000 ways of dialing the 4-digit number. But only one of those is specific to your bank card. So if you only have 3 trials, is it more likely than not, or less likely than not, that by a random search you'll be able to stumble upon the correct PIN number? Obviously, it is far more likely that you will fail by a random search. So that's the concept of specified complexity. Now, when we look into the distant past, obviously the past has already happened, and we can't repeat the past in a laboratory. And so how do we reason? How do we reconstruct what happened in the remote past? This is a very famous man in the history of science known as Charles Lyell, who was a famous 19th century geologist who wrote a very famous book in the history of science, a landmark classic called Principles of Geology. And in this book, Lyell insisted that if you want to explain changes in the remote past, changes in Earth's surface in the remote past, one should let one's present experience of cause and effect guide one's search for the best explanation. He pioneered what's called the historical abductive method of scientific reasoning. So to give an illustration, if you imagine that you're on an island and you discover volcanic ash, well, do you favor the volcanic eruption hypothesis, the earthquake hypothesis, or the flood hypothesis? Obviously, you would favor the volcanic eruption hypothesis, because in every realm of experience, volcanic ash comes from volcanic eruptions, not from floods or earthquakes. And so that is the principles on which the abductive historical methodology is based. So what are we trying to explain when we look into the distant past as what is of interest, what are we attempting to explain in the remote past, and why do we think intelligent design is in fact the best explanation? Well, at this point I want to introduce to you the idea that DNA in fact contains digitally encoded information content. Along the backbone of along the spine of the DNA molecule, along the sugar phosphate backbone, are chemical subunits represented by the letters A, C, T, and G. And the sequential arrangement of those characters basically allows the cell to function, basically determines what's going to happen in the cell. And These chemical subunits basically function like alphabetic characters in a written language or like digital characters in a machine code. The information in DNA directs the construction of proteins. The DNA is taken and it goes through a process known as transcription, and there's an intermediary molecule called messenger RNA, which is single-stranded, unlike DNA, which is double-stranded, and it's subsequently taken to a ribosome and translated into proteins. Proteins are the building blocks of life. And I'm going to show you a short animation by a colleague of mine, Dr. Stephen Meyer, who's providing the commentary for this visualization, which shows how this happens.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Was a trailer that was produced for Stephen Meyer's book, Signature in the Cell, which came out in 2009, which I highly commend to your reading. It looks at the origins of life and what best explains the origins of life. But as you can see in that animation, that visualization, you have in life an information storage, processing, retrieval apparatus. And that is something which in every realm of experience we habitually associate with intelligent designers, not with random processes or unguided processes such as evolution. Now, the genetic code is like a language system of life, if you will. You have, as I mentioned, as you saw in the animation, the messenger RNA is taken to the ribosome, and there are 3-character words called codons. And these 3 character words, there's 64 of them, and each of them corresponds to an amino acid. There's 20 different amino acids used in life. And so you have a language system, a codon to amino acid mapping system, if you will. And as you saw in the animation, the sequential arrangement of the amino acids determines how a protein folds. Amino acids are the subunits of proteins, and proteins might form jobs such as in biochemical pathways or in molecular machines. And you have different levels of structure of these proteins. The primary structure is the sequential arrangement of amino acids. The secondary structure is what is called alpha helices and beta strands, which together form beta sheets. Then you have the tertiary structure, where these come together to form what's called domains. And then the quaternary structure is where you have a multi-domain protein with proteins that possess multiple domains. Now, one question that we can ask, intelligent design and Darwinism make competing predictions on this, is how prevalent are functional folds in combinatorial sequence space? Let me explain what I mean by that. So, in the English language, there's an astronomical rarity of functional arrangements of alphabetic characters relative to non-functional arrangements of alphabetic characters. That's why the English language conveys meaning. And it turns out that in life we find proteins which are comprised of subunits which function like letters, and there are 20 different amino acids used in cellular proteins. That means that for a molecular length protein of 150 amino acids, there are 20 to the 150th power of possible ways of arranging those amino acids. That's 20 times 20 times 20 times 20, all the way up to 150. So huge number of ways of arranging those. You can call it the combinatorial space. So how prevalent are functional protein folds in combinatorial space? Under a Darwinian model, you would expect there to be a relative abundance so that you could find those functional folds by a random blind search. On an intelligent design model, you might expect that you'd find an astronomical rarity, which would evidence specified complexity and thus warrant a design inference. There was a paper published in this in 2004 by Dr. Doug Axe, who at the time was a postdoc at the University of Cambridge, and he published this in the Journal of Molecular Biology, and where he attempted to elucidate the answer to this question, looking at a particular enzyme called beta-lactamase, which is an enzyme involved in conferring antibiotic resistance in some bacteria. And he looked at a particular domain of this protein, which is comprised of 150 amino acids. And he employed a technique known as site-directed mutagenesis to randomly alter the protein fold, the amino acid composition. And according to Doug Axe, the prevalence of functional folds is astronomically rare. Let me now show you a brief section from a documentary produced by Illustra Media on this problem.
Richard Dawkins, the famous Oxford evolutionary biologist, has illustrated how the Darwinian mechanism works using a metaphor he calls climbing Mount Improbable.
From the front side, the mountain is a sheer cliff that could never be scaled in one giant leap. For Dawkins, this represents the impossibility of creating a complex animal by chance alone. Yet Dawkins also envisioned an alternative route up the backside of Mount Improbable, a long, gradually sloping trail of small steps leading all the way to the summit.
According to Dawkins, that's how you'd climb the mountain. And that's also how you'd build a Cambrian animal, one small step at a time. What chance alone can't accomplish in one blind leap, natural selection can accomplish through the cumulative effect of many small incremental steps.
In theory, each step corresponds to a small unit of biological change, a new gene and its protein product. But do mutations and natural selection have a reasonable chance of producing even one protein in the time available? Since 1992, molecular biologist Doug Axe has examined this question.
There's a story that's being told, and there's an appeal in the case of Darwinism to random mutation and natural selection as being, in vague terms, the mechanism. But if you look at the detail, what kind of mutation can accomplish these transitions? And there, it's important to realize that the one area where we can really nail this down is at the single protein level, where you can actually measure it. And if you look at protein structures, to get a substantially new protein fold is prohibitively difficult.
Each of the thousands of different proteins in nature is actually a chain made from a specific combination of 20 different amino acids. The sequential order of these chemical building blocks is crucial, for if they are arranged correctly, the chain folds into a functioning three-dimensional molecule. But if the amino acids are incorrectly assembled, no protein will form. If proteins are indeed rare among the possible sequences of amino acids, what are the odds that mutations would stumble upon a functional combination of chemicals from the vast number of alternatives? To find out, Axe randomly altered the structure of an enzyme protein comprised of 150 amino acids.
You've got the protein 150 amino acids long, then you've got 20 to the 150th power possible ways of arranging the amino acids. Out of all those possibilities, how many are functional and how many are gibberish?
If you do the experiments and you analyze how much information is required to get, say, a new protein fold, it's just far beyond what you can get by random mutation and natural selection.
How far beyond? Axe published his findings in the Journal of Molecular Biology. He determined that among all the possible amino acid combinations, the probability of generating just one short protein by mutation is roughly 1 in 10 to the 74th power, or 1 chance in 100 trillion, 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 trillion.
To put that in context, there's only 10 to the 65 atoms in the entire galaxy. So to build a new functional protein by selection and mutation within the time allowed for the Cambrian explosion, what you're essentially having to do is equivalent to a blindfolded man looking throughout the entire galaxy for one marked atom. So what we're talking about is searching for a tiny, tiny needle in an enormous haystack and having a very limited time to search.
So on the question of something like the Cambrian explosion, there does not appear to be any way that unguided random mutations can accomplish what needs to be accomplished to explain new functional proteins. And certainly by extension, wherever in the history of life you would need to have multiple new protein folds, the probabilities multiply. So there's no reason to think that this is plausible.
So the Cambrian explosion this documentary was about, which is an abrupt emergence of animal phyla about approximately 530 million years ago. I'll talk about that very briefly in the next presentation. One objection that's often raised is this idea of exon shuffling, which I'll explain. Domain shuffling is the hypothesis that you can generate new protein folds by recombining already existing domains. And this is thought to be accomplished by moving exons, that's protein coding regions of DNA, from one part of the genome to another, a process known as domain or exon shuffling. And there's different mechanisms by which this can be accomplished. There's a number of problems with this, however. First, the model at its core presupposes the prior existence of protein domains. You don't get beta sheets and alpha helices, which is the secondary level of structure, floating about on their own. The domain is the smallest unit of structure that you can get, and so it presupposes the prior existence of protein domains. And the model also assumes the ability of blind evolutionary processes to account for a specific toolbox of domains that can be recombined in various ways to yield new functions. And finally, the domain shuffling hypothesis in many cases requires the formation of new binding interfaces, so there's various problems with that rejoinder. As you heard, this represents a probabilistic resource problem. There are 10 to the 80 subatomic particles in the universe. Since the Big Bang, roughly 10 to the 16 seconds have elapsed. The time it takes light to traverse the smallest distance is 10 to the minus 43 seconds. By multiplying together the three relevant factors, we can determine that the maximum number of events that could have taken place in the observable universe since its origin is 10 to the 139th power. Any probability that goes below this, 1 10 to the 139th power, you can rule out the chance hypothesis. I think you can even do that at far, far lower levels. But this sets us a universal probability bound, if you will. And then we heard about the needle in the haystack problem. So the problem extends deeper, though. When molecular machines require multiple cooperating proteins in order to perform their function, the probabilities multiply exponentially. You can see there the DNA replication machinery involving a concert of machines which have to be crafted in very mutually specific ways in order to perform an overall job in the cell. But you might say, but there's still a chance, right? And this is what I call the dumb and dumber response. Here is a very well-known clip from a TV show in America where this man is asking this young lady, what are my chances? And she says, not good. And he says, not good like 1 out of 100? More like 1 out of 1,000,000. So you're telling me there's a chance. So that's what I call the Dumb and Dumber response to this. Then I want to introduce to you the problem of molecular machines in the cell and why this evidence is designed. Let me just show you a short guided tour of the world of the cell.
I'm finally just beginning to grasp the complexity of the cell. Are there systems within the cell that go well beyond Darwinian evolution? Some type of cellular technology that drives adaptation, replication, quality control, and repair? What if these new mechanisms have massive design implications? Well, I say, so be it.
So that's taken from an animation that was featured in a movie called Expelled, which documents many cases of persecution and intimidation that has been faced by intelligent design advocating scientists. I want to show you another animation. This is also produced by Discovery Institute, which is the main central think tank behind the intelligent design movement based in Seattle, Washington. This is like the power plant, if you will, of the cell.
It's been called one of the wonders of the molecular world, an amazing nanoscale machine. ATP synthase is a high-tech micromolecular power generator inside the cells of your body. It generates adenosine triphosphate, or ATP, an energy molecule that provides fuel that every one of your cells needs to function. Without this fuel, your cells will cease operation, and so will you. ATP synthase works like a rotary engine. The barrel-shaped rotator is composed of 10 to 15 protein parts called subunits. The rotator spins around, transmitting mechanical energy into the drive shaft of the machine, which helps make ATP. This drive shaft has a specially placed bump that opens and closes parts as the drive shaft spins around. This bump opens special protein subunits on the bottom of the machine. When the bottom subunits open, a spent energy molecule called adenosine diphosphate, or ADP, enters the machine. The mechanical motion causes the ADP to bind with an additional phosphate group, creating the ATP energy molecule. And the ATP drifts off into the cell, ready to power some biomechanical reaction. The ATP synthase machine has many parts we recognize from human-designed technology. A rotor, a stator, a drive shaft, and other basic components of a rotary engine. The ATP synthase is one of thousands of elegantly designed molecular machines inside your cells that make your life and all known life possible. ATP synthase, an example of intelligent design.
And the world of the cell is chock full of these nano machines that are operating on a nanoscale and operating with very extraordinarily high energy conversion efficiencies. The world of the cell is an absolute parallel universe, if you will, governed by molecular machines and information. I want to introduce to you now this concept of irreducible complexity in relation to molecular machines. Charles Darwin in The Origin of Species put forward a test for his theory that could potentially falsify his theory of evolution by natural selection. In The Origin, he wrote, "If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down." Michael Behe, the University of Lehigh in Pennsylvania, came along and put forward this idea of irreducible complexity, seeking to take up Darwin's challenge. He writes in his book, published in 1996, Darwin's Black Box, "By irreducibly complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning." He gave the example of a mousetrap that we recognize in our everyday experience. A mousetrap typically has a platform, has a spring, has a hammer, has a hold-down bar, has a catch, it has staples, and if you are missing just one of these pieces or two of those pieces, the trap doesn't work half as well as it used to, or a quarter as well as it used to. It's broken. And so how would you build that up by virtue of numerous slight successive modifications? That seems to be a problem. And moreover, the trap still might not work if the holding bar is too short or the spring too lightweight. And so there's all kinds of interface compatibility issues as well. He put forward the bacterial flagellum is one of his favorite examples in the book. And the bacterial flagellum has kind of become the mascot or the flagship of this concept of irreducible complexity. The flagellum requires a minimum of 3 parts to work: a rotor, a motor or power source, and a paddle. If any of those parts is missing, the flagellar motor does not work. This is a rotary motor engine that propels bacteria through a medium. They have a very high energy conversion efficiency of virtually 100%. And in some bacterial species like Vibrio cholera, these things are spinning at speeds of up to 100,000 RPM. Salmonella, they can spin at speeds of up to 18,000 RPM. And they're plugged into an elegant signal transduction apparatus, so they're getting feedback from the environment, and very, very complex. They actually build themselves from the inside out. Very, very sophisticated molecular machines. Now, why is irreducible complexity evidence of design? Explaining the origins of irreducibly complex features requires a cause that possesses foresight. Only intelligence can visualize complexity and bring together everything that is necessary to actualize a complex endpoint. Finally, then, what are some of the common objections to intelligent design? One of them is that it's a God of the gaps objection. So the God of the gaps objection basically says intelligent design is used as a placeholder for that which we're ignorant of. And as we progress in science, as we make new scientific discoveries, intelligent design has to retreat with each new advance as we learn more and new gaps in scientific knowledge are filled. Whereas, as I've shown earlier in the presentation, intelligent design really is not an argument from ignorance, but a positive inferential case for design. It's based not on what we don't know, but what we do know about the cause and effect structure of the world. The fact that information is uniformly associated with conscious activity, with conscious, rational, deliberative agency, intelligent design. Another objection is that intelligent design isn't science. This is one that we hear very often. And there's a number of reasons that people say that intelligent design isn't science. We hear that intelligent design isn't science because it violates methodological naturalism. Whereas actually intelligent design is a natural phenomenon that we have experience of, and we experience intelligent design in the natural world. So intelligence is a natural phenomenon. Intelligent design as a theory doesn't necessarily commit you to the supernatural. So I don't think it does necessarily violate methodological naturalism. It certainly violates methodological materialism, because consciousness is not material, I would argue. Sometimes people argue intelligent design isn't science because it's not falsifiable. and falsifiability was originally put forward by a famous philosopher of science called Karl Popper, and it's the idea that something counts as science if and only if it can be falsified somehow by running an experiment which shows it to be wrong. Karl Popper's criteria of falsifiability, however, for demarcating science and non-science has been widely abandoned by professional philosophers of science because it's fraught with problems and difficulties. For example, scientific paradigms typically are not refuted by a single empirical experiment, but rather it takes a cumulative case to eventually overhaul a scientific paradigm. So one single experiment usually doesn't refute a well-worked-out scientific model. In any case, intelligent design, I would argue, is falsifiable. Show either that life doesn't contain specified complexity, or that specified complexity has a better explanation that can be explained by natural processes, then intelligent design as an inference would be falsified. Another objection is intelligent design doesn't make testable predictions, whereas I would argue that it actually does make testable predictions, because it predicts that when we look at the cell, we will find specified complexity, the hallmark of design. Another argument is intelligent design isn't science because it has no heuristic value. Heuristic value is, what value does it have in progressing science further? So it's argued as a science stopper. Whereas I would argue, actually, intelligent design encourages us to look for functions, for example, for junk DNA, whereas the Darwinian alternative might encourage us not to look further for functions. Or for apparently suboptimal design. Why is it designed the way it is? So I think intelligent design does in fact have heuristic value. Another objection is intelligent design isn't science because it invokes unobservable entities, whereas other disciplines like particle physics also invoke unobservable entities, so that won't do either. Or intelligent design isn't science because it doesn't address the modus operandi of design. How did the design come to be implemented? But we don't even understand today how consciousness interacts with the material world, and yet we know that intelligent agency is a real phenomenon in nature. And supposing a group of astronauts were to find a hieroglyphic inscription on the backside of the moon, they might have no idea how it came to be there, or what types of tools that were used, but nonetheless they would be warranted and quite justified in inferring design. Finally, intelligent design isn't science because a judge in Dover said so, which of course refers to the 2005 Kitzmiller v. Dover ruling, where a district judge ruled that intelligent design is not constitutional because it's not science. But since when was it the place of a district judge to make pronouncements concerning the philosophy of science? Then we have the argument from suboptimal design, the idea that some systems in life seem to be suboptimally designed or designed poorly. This is the inverse wiring of the vertebrate retina. Over here, in vertebrates, the retina seems to be wired the wrong way around, which is supposed to be a suboptimal case of design. And here we have the recurrent laryngeal nerve, where the laryngeal nerve loops around the aorta on the way up to the larynx, and it's supposed to take a circuitous route. And so it's thought that this is a case of suboptimal design. Whereas I would like to put it to you that actually this argument from suboptimal design is itself not a God of the gaps argument, but a Darwin of the gaps argument. Because it has to retreat with each new advance in scientific discovery. As science advances and we learn reasons why it's designed the way that it is, this argument has to retreat. And in fact, many cases have been found where there is plausible function for why it's designed the way it is. And moreover, if you're an intelligent design proponent and you accept common ancestry, then there might be constraints placed upon these features by the ancestral pathway that's been taken. So in light of those arguments and others, I'm not persuaded at all by the suboptimal design argument. We even find cases of suboptimal design in human engineering and design. So it's not a case against intelligent design simplicator at all. So finally then, let's draw some conclusions. Intelligent design is based on standard principles of scientific reasoning about the past. It's based on the historical abductive method. Intelligent design challenges the concept of unguided chemical and biological evolution. The design inference is based on cutting-edge advances in molecular biology, which reveals the complexity, elegance, and engineering prowess of cellular machines. Thank you.